Hello, everyone, and welcome to our first lecture of our new 15-week MCAT prep course. So the first lecture today is going to be focused on cell theory and cell development. All right, so this is going to be a question-based structure. So basically what we're going to do is there's going to be about 10 questions per lecture. And you'll have a couple minutes to answer each question. And then after, I will go through all the topics that were covered in each question. And yeah, that's how it's going to go. So here I linked our GroupMe, our Instagram, our YouTube channel, and the schedule is on here. So every Sunday at 8:30 p.m., we will have a recorded lecture that's released. And then Mondays, it will be live and on Zoom, and the Zoom link will be sent as a link in that GroupMe. So definitely get in that GroupMe as soon as possible. All right, so our first question is going to be about cell theory. And the question states, which of the following is a premise of cell theory? 1, all living organisms are composed of one or more cells in their products. 2, all cells arise from pre-existing living cells. And 3, all living cells require water for survival. So now you can take a minute, pause the video, try not to take more than 2 minutes to answer the question. And just restart when you answer it. All right, so assuming now that you've answered the question, I'm going to show the answer, which is C. So 1 and 2, those are 2 out of the 3 premises of cell theory. So usually the way that you would tackle a Roman numeral question is by finding which number shows up in exactly 2 of the answers, because that will help you cancel out 2, and it's just the fastest way to do these Roman numeral questions. Unfortunately, here in the answers, there are none that show up in exactly 2. So you could really just start with any and go by process of elimination that way, just to narrow down your answers. If you're certain about one number, then you know that you can cross out a couple other ones. All right, so the premises of cell theory, there's 3. So these were defined by Robert Hooke. So the 3 premises are that all living organisms are composed of one or more cells and their products. Cells are the basic structural unit for all organisms, and all cells come from pre-existing cells. So it's pretty basic, but that's really all that you need to know for the MCAT about cell theory. So on to question 2. So this question, which of the following is not considered a living organism? So prokaryote, cyanobacteria, eukaryote, or virus. Again, take a minute, pause here, take some time to answer the question, and resume the video once you do that. All right, so assuming that everyone's answered the question now, I will reveal the answer, which is D, a virus. So actually, you should be able to cross out A and B right away, because cyanobacteria falls under the category of prokaryotes, so it can't. 2 answers can never be correct on the MCAT, so you know that those can be crossed out right away. And then eukaryote, that's like a normal animal cell, so we know that that is considered a living organism, so virus is the correct answer. So now I'm just going to review the differences between those 3. So this is a pretty general overview. We'll dive more into prokaryotes specifically later in today's lecture, and eukaryotes we'll talk a lot more about in a cell structure lecture. I think that's week 6, maybe. It's in a couple weeks. But anyway, so prokaryote actually means before the nucleus. So that's a good way to help you remember that prokaryotes don't have a nucleus, and they don't have any membrane-bound organelles. So that's basically the defining difference between prokaryotes and eukaryotes. And they're also evolutionary, the oldest domain. So since they're before the nucleus, it just helps you remember that they're really, really early on is when they evolved. So they reproduce asexually. There are a couple different ways that they do that, which we will go over a little bit later in today's lecture. So prokaryotes include bacteria, which is the most common, and that's what people generally will think of when you think of prokaryotes. So it's just a most commonly a unicellular microorganism that has a peptidoglycan cell wall. There are some other components of cell wall, and there are a few differences in different types of prokaryotes. And again, we'll talk about that in depth a little bit later. So there's also archaea, which are single-celled organisms. They're very similar to bacteria, but these are found at really extreme pressures and temperatures. So things that live near like vents on ocean floors. So where normal living organisms can't survive. So really, really hot, really, really cold. These are also called extremophiles, which is definitely helps you remember that because they live at the extremes that normal living organisms can't tolerate. And then there are cyanobacteria, which are microorganisms that are capable of photosynthesis. All right, so eukaryotes, this is the classic cell picture that I'm sure you're all very familiar with. So the eukaryotes contain membrane enclosed organelles. So they have a nucleus, mitochondria, all that good stuff. And we will go over cell structure in depth again. I think it's week 6. So in a couple weeks, we will go through all these structures, but I have listed the ones that you need to know for the MCAT here. So that's the nucleus, mitochondria, ribosomes, rough ER, smooth ER, Golgi apparatus, lysosomes, and peroxisomes. So if you want to start familiarizing yourself with those, that would be great because those are definitely pretty high yield on the MCAT. So these can reproduce either sexually or asexually. So meiosis is the means of sexual reproduction and mitosis is asexual. So eukaryotes obviously can be either single or multicellular. Obviously we are multicellular creatures, but they can survive on their own as a single-celled organism. So viruses are not considered living organisms, and that's because they're obligate intracellular parasites. So they need a host cell's machinery to reproduce and to function. And when they're by themselves, they have no activity. So that lack of activity on their own is what makes it so that they can't be classified as a living cell or a living organism. And there will be a question on viruses at the end of today's lecture. So we'll talk a little bit more about how they reproduce. All right, question 3. So now we're going to dive in a little bit more about prokaryotes. So which of the following is associated with prokaryotes and introduces new genetic material into the cell? So we have binary fission, transduction, conjugation, and mitosis. So again, here you can take a minute, pause the video and answer the question. All right, so assuming that you've answered the question now, I will show the answer, which is C, so 2 and 3. So transduction and conjugation both are associated with prokaryotes and introduce new genetic material. So again, a Roman numeral question. What I would encourage you to do is find which one is included in exactly 2 answers. So for this question, that would be 1, binary fission. And binary fission does not introduce new genetic material. It's basically when a cell grows and just divides in half. So it's a asexual reproduction and involves no outside genetic material entering the cell. So you can cross out 1 right away. So that gets rid of A and B. Now you're left with C and D. And the only difference between those is number 4, mitosis. So mitosis is not associated with prokaryotes, so we can get rid of that one pretty quickly. And then we're left with transduction and conjugation. So even if you're not sure what transduction and conjugation are, by using this method, you could still get to the right answer. So definitely would encourage you to familiarize yourself with that strategy when attacking these Roman numeral questions. And they're pretty common on the MCAT in all the sections. They love the Roman numerals for some reason. All right, so transduction. So transduction is the transfer of genetic information from one bacterial cell to another via lysogenic phage. So that's a virus. So this shows kind of a viral reproduction cycle, but also how that works to transfer DNA from one bacteria to another. So the virus will go in, lyse, or yeah, like basically just cut up the host bacterial cell DNA. And then it will use the those available nucleotides to make its own DNA so it can reproduce, and it uses the machinery present inside of that cell to make copies of itself. So again, that shows how the virus is not a living organism because it's using another cell, a living cell, to make copies of itself. So this is not like an intentional kind of reproduction. So yeah, what I have on here is it happens by accident. So basically when the virus is recondensing its own genetic material in that capsid, sometimes fragments of the host DNA can just get mixed up in there. So, and then it will be carrying that host DNA when it goes and infects a new cell. So when it infects that new cell, sometimes it can insert that DNA into their DNA, and that's just a way for genetic diversity to happen within prokaryotes. The transformation, this is not super common and it's pretty low yield, but I just wanted to include it so you're familiar with it. So it's the uptake of genetic material from the extracellular environment. So basically like a plasmid, if that enters a cell, then homologous recombination can introduce that into the bacterial cell's genome. We'll talk a lot more about DNA and recombination and things like that in a couple of weeks. All right, so conjugation. So this one's pretty important, pretty high yield, I'd say, for in terms of like prokaryotes and microbio. So conjugation requires, so this is still asexual. I know it's kind of tricky, but this is still considered asexual reproduction. So basically you have a F positive or F negative cells. So F positive cells are considered male. And they contain the F factor, which is an extra chromosomal element. So it's not in the DNA, but it's basically like a little snippet of DNA that's not present in the rest of the DNA. So conjugation can only occur between F positive and F negative, or HFR and F negative cells. So two F positives can't, conjugation can't happen there. Two F negative cells can't happen there. Two HFR cells can't happen there. So it's F positive or HFR and an F negative. So HFR basically just means that this extracellular component has, or extra chromosomal, component has inserted into the DNA. So that just means it's high frequency recombination. So again, we'll talk a lot more about recombination in a couple of weeks when we talk more about DNA replication and things like that. But anyways, a little bit more on the next slide. So the F factor encodes for a sex pili, which basically makes a bridge between the two cells, as you can see in the picture over here. So when it makes this little bridge, that allows a copy of the F factor to be inserted into the previously F negative cells. The end result of this is two F positive cells. Yeah, so this is pretty important. So I would take some time to just familiarize yourself with the difference between F positive, HFR, and F negative. So binary fission, this does not introduce new genetic material. So this is probably the most common form of reproduction for prokaryotes that will be seen on the MCAT. So this just results in two exact copies of the original genome. So it's two identical daughter cells that are identical to the parent cell. So basically, you can think of it kind of like the bacterial version of mitosis. Basically, the cell grows big enough until it has enough machinery for two distinct cells, and then it divides in half, leaving two identical copies. So this is an exponential type of growth. So as you can imagine, when you start with one cell, it takes a certain amount of time, divides, now you have two. Both of those, certain amount of time, divide, now you have four. All four of those take a similar amount of time. Yeah, so basically it's an exponential type of population growth until obviously nutrients are depleted and they can no longer grow. But anyways, the exponential phase of growth is known as the log phase. So that's pretty important to know. All right, so now we're going to talk a little bit about some different classifications for prokaryotes. So question is, you have an unknown rod-shaped bacteria that turns dark purple with gram staining. How would you classify this bacteria? So take a second here, pause the video, and resume once you've answered the question. All right, so assuming that you've answered the question now, I will show the answer, which is A, it's gram-positive bacilli. So we'll talk about what gram-positive versus gram-negative and all these different shapes here too. All right, so these are good to know for sure. And I have pictures on the following slide. So round bacteria is classified as a cocci. Honestly, I'm not sure how you say it. But anyways, the way I remember that, it's pretty elementary, but just looking at the word, like all the C's and O's, they're very round, you know, they all look like little circles. So you just remember that that's a circle. Similar way of remembering rod-shaped bacilli. So all the I's and L's right there, they're all long, skinny, look rod-shaped. So might not work for you, but that's what worked for me for remembering these. And then the spiral shaped ones are pretty easy to remember because it basically says the word spiral in the name. Yeah, so there are a few more subclassifications of that. So, Coccus is a singular one. Diplococci is 2. Staphylo is a bunch. Strepto is a line. Yeah. So I would just be familiar with this. Don't spend hours memorizing all of these. Not super high yield, but definitely good to at least be familiar with. Bacilli, so it's a similar thing. So there's a coccobacilli, which is basically a cross between a cocci and a bacilli. So again, you have diplo, that just means 2, streptos in a line, and then palisades. I don't know, honestly, wasn't super familiar with that one myself. It's definitely not a super high yield thing to know, but again, just familiarize yourself with this. Look over these images. So gram staining. This is definitely important to know, and it also tells you a lot about the different structures of prokaryotes. So it's basically a way to classify bacteria based on the structure of their cell wall. So if it stains strongly, it'll turn dark purple, and if it stains weakly, it turns light pink. So gram-positive, this one will stain strongly. So these will be dark purple. So they have a thick peptidoglycan cell wall outside of the cell membrane. So they're more easily targeted by antibiotics because they don't have an additional membrane on the outside of their cell wall. So a lot of antibiotics will target the cell wall and like poke holes in it to disturb osmotic pressure and things like that. So these gram-positive organisms lack an additional outer cell membrane, which is different from the gram-negative, which have, so the main difference is, one, their peptidoglycan cell wall is much, much thinner, as you can see from these 2 pictures, and they have an additional cell membrane, which provides another layer of protection. So you would need a different strain of antibiotic to target the membrane or something that can diffuse across the membrane and then target the cell wall. So these are usually more resistant strains. All right, question number 5, nutritional classifications. If a bacterium has infected the human bloodstream, which classification is it most likely to fall under? Chemoautotroph, chemoheterotroph, photoautotroph, or photoheterotroph? Take a minute, pause the video, and we'll go over the answer in a minute. All right, so the answer is chemoheterotroph, and we will discuss this. All right, so autotroph versus heterotroph. So autotrophs utilize CO2 as their carbon source, and they produce their own food for energy. So you can think of like plants doing photosynthesis, they would be a photoautotroph. Yeah. And then a heterotroph, you can think of just like normal mammals. They rely on other organisms to get their organic nutrients, and they cannot produce their own food from energy. So chemotroph get their energy from chemicals. So like, again, just like eating something, and then phototroph get their energy from light. Again, most easily you can think of as plants. Some bacteria like the cyanobacteria do this as well. So I just have these all broken down here. I'll go over them quickly. So chemoautotrophs, they build their own organic molecules from carbon dioxide by oxidizing other inorganic molecules. Chemoheterotrophs require organic molecules made by other organisms as their source of carbon. Photoautotrophs use CO2 as their source of carbon, obtaining energy from the sun, and photoheterotrophs require an organic molecule from another organism as their carbon source, but they get their energy from the sun. So I think photoheterotrophs is probably the least common, but it's still there. It's still a possibility. All right. Question 6, eukaryotes. PCD, also known as primary ciliary dyskinesia, is an inherited disorder that results in immotile cilia. This disorder causes a protein deficiency in which of the following structures? Take a minute here, pause the video, and answer the question. All right. Assuming you've answered the question now, the answer here is A, microtubule. So we'll go over all of these except for Golgi apparatus. If I was answering this question on the MCAT, that would be an answer that I would quickly just throw it away since it's not related to the other answers. So that's usually a pretty good sign that it's not going to be your answer. Yeah, so our 3 types of filaments on the MCAT that we talk about: microtubules, microfilaments, and intermediate filaments. So microtubules are the largest of the 3, and they're composed of alpha and beta tubulin. So the alpha and beta tubulin will form basically a sheet which like rolls into a big tube. So microtubule, think it's actually like tube shaped. So these are anchored at microtubule organizing centers, which I also call the MTOC, which is probably what I'll call it from now on. So they're anchored there. So microtubules are known for their very dynamic structures. So the alpha and beta tubulin can rapidly be added on or come off. Yeah, be added on or come off, allowing it to grow and shrink and be like very dynamic structure, but it is not dynamic from the MTOC end. It's got a strong anchor there. So that's definitely important to know. So these mediate transport within the cell, and they are the structural unit of cilia and flagella. So that's why microtubules was the answer for this question, because if you have a disorder in cilia, then it's the microtubule, most likely. That is the root of the problem. So microfilaments, these are the thinnest of these 3 types of filaments. So they're composed of actin. So we'll talk more about this when we talk about like muscle contraction. But anyway, so they're responsible for gross movements of the entire cell. So they're also very dynamic. They can grow and shrink with the addition or removal of actin. So they disassemble and reassemble as needed by the cell. So intermediate filaments are, their size is in between that of microtubules and microfilaments, which is pretty explanatory in the name. So these are composed of a wide range of polypeptides, and they provide rigid cell structure. So they help the cell resist mechanical stressors and things like that. So intermediate filaments are more different, and microtubules and microfilaments have some similarities. The intermediate filaments are not dynamic, they're pretty static, and that helps them be a little bit more sturdy. So here I just have a picture. this is definitely, it's helpful to see it like this. It helps you remember sort of the shape. Here you can see in the microtubule how it forms that tube. So all these little circles here are the alpha and beta tubulin. Yeah, and then the microfilament too, you can see they're all actin. So cilia and flagella, we'll just go over these quickly. So cilia are small hair-like structures that help move fluids over the cell surface. So you can think of these as being these are in the respiratory tract and in the inner ear. So we'll talk more about their structure when we do a lecture on the respiratory tract and hearing. But just know that they're like little hair-like structures, and just think of this picture when you think of cilia. So flagellum is just one large tail. So it's important to know that distinction between cilia and flagella and not get those two mixed up. So when you think of flagella, the easiest way is to just think of a sperm cell, and that's actually the only human cell that uses one. So that's where that gets its motility. So yeah. All right, question number 7. So we're gonna go over the cell cycle. This is super high yield, so I would definitely take the time to familiarize yourself with the cell cycle and all the steps. And we're gonna go over that in just a minute here. All right, so a highly specialized cell is most likely to be permanently stuck in which phase of the cell cycle? So G0, S, M, or cytokinesis. All right, take a minute, pause the video, and resume once we answer the question. All right, I'm going to assume you answered the question now. So the answer here is G0. We're going to go over all the phases of the cell cycle now in depth. Again, super important to be familiar with all of these steps. All right, so here we have an overview of the cell cycle. So basically, inner phase is G1, S, and G2, and then the mitotic phase is mitosis and cytokinesis. So a cell will only enter mitosis when it's ready to divide. So some cells don't ever divide, like highly specialized cells that are not constantly replaced. So I'll talk about what kind of cells those are, but those are dormant, and they're dormant in G0. So basically they're not growing. They don't go to the S phase. G0 is like being stuck over here in G1, basically. All right, so inner phase, which is composed of G1, S, and G2. So G1 is growth phase 1. So cells that are permanently in this stage are dormant and are in stage G0. So was the answer to our question. Highly specialized cells like heart muscle cells and neurons, you're not constantly reproducing neurons. Like if you think about when you have nerve damage, you don't just regrow new nerves. Like that's usually a pretty permanent problem. So those cells are in G0 because they're not constantly growing, they're not synthesizing, or they're not making new things. They're there, they're doing their job, and they're not coming back if they die. So in G1, if a cell is progressing forward, this is where the cell will be making proteins and organelles that they need for cell division and for synthesis. So synthesis is the S phase, so that comes after G1. And at this point, the cell replicates its genome using those proteins that were just produced in G1. So in S phase, the centrosome also duplicates. So that's an important step to know for sure. And then G2 is growth phase 2. So this is where protein synthesis continues until you have enough organelles for 2 distinct cells. And then at the end of G2, there's an important checkpoint that scans for DNA damage before moving into mitosis. So I'm going to talk more about checkpoints in cell division when we talk about DNA synthesis, just because I thought it fit in a little bit better with that lecture. But just know that that's a really important checkpoint for the cell, because if there is DNA damage, then the cell will probably apoptose, which means it will die. as to not reproduce with damaged DNA and keep passing that on. So after interphase, we have mitosis, which can be the M phase. So this is where the cell is actively dividing. So mitosis produces 2 diploid daughter cells because they have 2 copies of their chromosomes. So that's diploid. Mitosis is divided into prophase, metaphase, anaphase, and telophase. Definitely need to know those steps and their order at the bare minimum. Also should really know what happens in each step, but most importantly, know the names of those steps and the order that they happen in. This is probably not super helpful, but the way that I remembered the order of the steps was just thinking like PMAT. For some reason, that just sounds right to me. Like I think because I had some engineering friends that took the GMAT after graduating, so I just hear PMAT, MCAT, like it just sounds like another standardized test. You know, anything else, any other combination just wouldn't sound right. But one mnemonic that I saw when I was studying was IP on the mat. So I is interphase, P is prophase, and then mat, metaphase, anaphase, telophase. So however you want to remember it works, but just thought I'd share. All right, so now we're going to dive a little more specifically into mitosis. So here we go. So the nuclear envelope is present in which of the following phases of mitosis? So anaphase, telophase, metaphase, or prophase? Probably not super helpful that I didn't put those in order for the answers, but that's all right. All right, take a minute here, pause the video, and answer the question. All right, assuming that you've answered the question now, the answer is telophase. So we will go through all of these, but you should know, you should be reminding yourself, using these questions as a chance to recall. What I found when studying for the MCAT is the best way that I could remember things was by forcing myself to do recall whenever I had a chance. So if I was reading one thing and I saw a reference to something else, I would try to pull that information out. I think just constantly doing any sort of recall that you can really helps solidify your knowledge of the material. All right, so again, I know we just talked about this, but I'm going to just reiterate one more time because I think it's really important. The 4 phases: prophase, metaphase, anaphase, telophase, PMAT. That needs to definitely be remembered. So basically, cells need to do this so they can replace lost or damaged cells. So you think about like every time you got like a cut on your finger if it never healed. So you need to be able to reproduce cells asexually just to survive. So these are not the super highly specialized cells that are going into mitosis, but a lot of cells in the body are not constantly, but fairly consistently dividing. So prophase, this is our first step of mitosis. So this is where the genome becomes visible, and you think of chromosomes in those nice little X-looking shapes. So this is where the genome is packed into chromosomes. So I bolded a bunch of terms on here. I think these are just like keywords that are pretty high yield for the MCAT. So being familiar with them and knowing which words to associate with which step of the cell cycle is super important. So basically each chromosome consists of 2 sister chromatids, which are the 2 copies of a chromosome. So in prophase, the nucleolus disappears. So this is why we don't have that nuclear envelope. It's degraded in this phase. And then we have our spindle and kinetochore fibers, and the centriole pairs begin to move to opposite ends of the cells called asters, which means like star, I think. So that's just due to their appearance because they kind of look like stars. So here in the middle, we have our chromosomes, and then we have our centrioles on the opposite ends of the cell. All right, so metaphase, you can think of, they all line up in the middle, metaphase middle. that's a good way to remember that. The metaphase plate is what it's called. So this is where the kinetochore of each sister chromatid is attached to spindle fibers on the MTOC of opposite ends of the cell. So the MTOC, again, that's the microtubule organizing center. So that's where the microtubules are anchored. And yeah, so that's the centrioles. It's essentially the same thing. So this is where they attach. So they're getting ready to pull apart, but they're not pulled apart yet in metaphase. Just think metaphase middle. They line up right there on the metaphase plate. All right, anaphase is our third step. So this is where the spindle fibers shorten. So they start pulling apart these sister chromatids, and this is where we start with the cleavage first. So that's when you start getting this little indentation that's indicating that the membranes are being pulled apart and you're starting to form two distinct cells. So again, here's an important indication for our microfilaments. So the microfilaments actually form a ring around the cell and they start shortening. So again, we have the importance of the dynamic ability of microfilaments to either lengthen or shorten as the cell needs to perform their function. So while they're shortening, they're starting to pinch off that cell membrane to create those two distinct daughter cells. So telophase here, we have that microfilament ring is closing and they're budding off to two distinct cells. So here we have the nuclear envelope reforms and surrounds the chromosomes. That was the answer to our question. This is where the nuclear envelope is reintroduced because it couldn't be present before because the chromosomes had to be out and lined up on that metaphase plate and do all that other stuff. So yeah, so once that nuclear envelope reforms, the chromosomes can decondense and they're just as free DNA. So the chromosomes aren't usually like packed up like how they are during cell division. So once they're in that nuclear envelope, they can decondense. Yeah, and then the cells pinch off and now you have two distinct daughter cells that are identical to the parent cell. All right, question 9 we have about meiosis. So what is the genetic makeup of the daughter cells after meiosis 1 is complete and before meiosis 2 has begun? So we have either 2 haploid daughter cells, 2 diploid daughter cells, 4 haploid daughter cells, or 4 diploid daughter cells. So just take a minute here, pause the video, and answer the question. All right, assuming that you've answered the question now, we can go over the answer. So the answer here is 2 haploid daughter cells. So haploid basically just means that they do not have 2 copies of the chromosome. So diploid, you think, I mean, diploid sounds like 2. So it's either half or not half. All right, so yeah, we'll just go over it really quickly here. So a diploid cell you think of as 2N. It's commonly referred to on the MCAT as either N or 2N. So this is where you have 2 complete sets of all of the chromosomes. So in mitosis, it's important to note that the resulting cells are always diploid. And then for meiosis, both meiosis 1 and 2, they're haploid. So the difference is at the end of meiosis 1, you only have 2 haploid daughter cells. And at the end of meiosis 2, you have 4 haploid daughter cells. So a few differences between mitosis and meiosis, aside from the number of chromosomes that you have. So mitosis is just the one phase, but meiosis is split into meiosis 1 and meiosis 2. And that is so you can have 4 daughter cells at the end. So it's basically like doing 2 rounds. Except in the second round, there's no chromosome duplication at all. So yeah, I think it would be probably a good idea to just look over this image here on the right and familiarize yourself again with what happens in each step. But since we just went over, I'm not going to go over it again. But definitely just take some time and look over this figure. So meiosis 1 and meiosis 2 are both pretty similar to mitosis. So it's the same 4 phases: prophase, metaphase, anaphase, telophase. but I just have a few differences that you should definitely be aware of. So one difference is that in metaphase 1, we have tetrads that align at the center of the cell and not sister chromatids. So it's the tetrads that will be pulled apart, not the sister chromatids won't be pulled apart. So it is important to note that after telophase 1, the 2 daughter cells are both haploid. So at the end of telophase mitosis, it's 2 diploid. So meiosis 2 is actually identical to mitosis, except for the fact that the starting cells in meiosis 2 are haploid. So again, I have another image here. Definitely just take time, look over this figure. You know, I find it helpful to have like a visual reference for things like this. Sometimes just reading it, it's just a lot of text. So being able to picture what the chromosomes are doing, where they are, what the cell's doing, is definitely helpful. Skip the slide. All right, question 10. So we're going to talk a little bit about viruses. So the question is, a scientist adds a drop of an unknown substance onto an agar plate with E. coli. The following day, there's a clear spot on the plate where the substance was added. The substance could be A, a virus undergoing the lytic cycle, B, a chemical that is toxic to eukaryotes, C, a virus undergoing the productive cycle, or D, a bacteria undergoing transformation. So just take a minute, pause the video, and take some time to answer the question. All right, assuming you've answered the question now, we can go over the answer. So the answer is A, a virus undergoing the lytic cycle. So we're going to talk about all these, the different cycles of reproduction for viruses, but I'm just going to go through some of these really quickly. So just looking at the question, you could cancel out B because it's talking about eukaryotes, and we're talking about E. coli, which is a prokaryote. So something that's toxic to eukaryotes, we can't definitively say is also going to be toxic to prokaryotes. So B can be canceled out. And then D, also bacteria undergoing transformation. That's kind of irrelevant to the question in general. There's no reason to think that that would make a clear spot on the plate. And then C, a virus undergoing the productive cycle, can be our answer because the productive cycle pertains to animal viruses. And we're talking about prokaryotes, not eukaryotes. So the only possible choice left is A. All right, so the lytic cycle. So the lytic cycle kills the cell immediately. It goes, not immediately, but once the virus is in, the cell's not going to survive. So basically the virus will enter the cell, and then it degrades the host genome, uses host machinery to replicate its own genome, and produce capsid proteins, which are what, like this little like head part is called a capsid. So it also uses the dNTPs from the degraded host genome in the synthesis of its genome. So basically, these capsids will automatically assemble around the replicated genomes and start reforming that virus. So at the end, one of the later stage genes will produce an enzyme called lysozyme, which will degrade the bacterial cell wall. So by doing this, it disturbs the osmotic pressure of the cell. So without that rigid cell wall to hold it in, the cell membrane will burst and it'll lyse the cell. So this will release however many copies that virus made within the cell, they will be released and they are able to go infect other cells. So this is a good way for viruses to really, really rapidly increase their numbers. This is pretty quick reproduction and distribution of the viruses. So the lysogenic cycle, this actually ends up going into the lytic cycle, but not initially. So initially the virus's genome will be inserted into the host genome. So it will stay dormant until the host reproduces. So the virus doesn't go in and immediately do anything. It just goes in, inserts into the host genome, and it's copied. So eventually, once it keeps dividing, the prophage will be activated, and then it will remove itself from the host genome, and then it enters the lytic cycle. So both the lysogenic and lytic cycle eventually will end up killing host cells, but lysogenic doesn't do that initially. Initially, it's dormant for a while. So you can be infected with one of these and not know for some time. So this is, again, earlier we talked a little bit about transduction. So this is like when the viral DNA excises itself from the host genome. Sometimes there can be errors that take some host genome with it, and that's a way that it can introduce new bacterial DNA into a new cell when it goes and infects that next cell and inserts into that DNA. So I just have a picture of that here. So I think it would be a good idea to familiarize yourself with this. Look over this. So you can see down here in the bottom part, this looks exactly like our lytic cycle, and that's because it is. So the productive cycle, this only pertains to animal viruses because it plays off a component of the cell membrane. So you can't do that in a prokaryote when you have that thick cell wall there, because that will inhibit this budding that's necessary for the productive cycle. So this is the only one where the cell will not die. The host cell doesn't necessarily die because the virus is able to bud off. So a component of the cell membrane, which we'll talk more about when we talk about cell transport, is endocytosis and exocytosis. So it can basically form these little buds, and the cell membrane just closes up right behind it. So it doesn't leave any holes. It doesn't disrupt the cell membrane in any way. Yeah, it's definitely a really key component of the cell membrane. Just like when you think about cell division, when they pinch off, there's no hole there. It's not like leaking organelles out because the cell membrane is able to just sort of pinch and be shut. All right, so that is all that I have today. Thank you for watching. And if you have any questions, feel free to just send a message in the GroupMe in general, or you can message me directly. Yeah, good luck studying, and I will see you guys tomorrow. Thank you.